Hi everyone, my name is Katherine Leip and today is June 26th and this is my Unit 4 presentation. So gene therapy is an upcoming and important part to biological and genetics research. So genetic modifications by exogenous DNA could possibly be a treatment for inherited human diseases like sickle cell and Parkinson's and Alzheimer's. So gene therapy is bringing a new treatment options to a variety of different medical fields. It's offered a theoretical advantage of different clinical benefits. Several categories that have been approached and listed throughout the article that I will touch on briefly, such as genetic engineering from viral vectors to genome editing, specifically looking at retroviral vectors and adeno-associated viral vectors, also AAV. So retroviral vectors was developed in the 1980s to the early 1990s. It showed to deliver genes into repopulating HSCs, which are C-type retroviruses adapted for efficient gene transfer into primary lymphocytes. And those were used in the first generation clinical trials to deliver normal copies of a specific defective gene into the genome of T cells. So AA vectors are basically they're engineered from non-pathogenic, non-enveloped parvovirus that is a naturally replication defective part. So AAV vectors are predominantly non-integrating. So that means that the transferred DNA is stabilized as a episome. This feature lessens the risk related to integration, but it also limits the long-term expression from the AAV vectors to the long-lived postmeiotic cells. So some of the data that was done through this was one of the clinical trials for hemophilia B. And that was basically started in the late 1990s, and it tested for AAV vectors to muscles via injections, and then moving to the intravenous administration. It was early, but it was also limited to insufficient dosing and anti-AAV immune responses. So looking at the genome editing, it was performed on cells of ex vivo or the editing machinery. It can be delivered in vivo to affect the in situ genome editing. So the genome editing landscape, it basically changed in 2012 with seminal discovery by Dunana and Charpentier, and both of those people showed bacterial defense systems that were composed of clustered regulatory interspaced short palindromic the CRISPR-Cas9 nucleases, and those are effectively programmed to cleave DNA at sites or the interest. So potential of genome editing strategies to bypass pathology and muscular dystrophy alters the splicing of mutated genes. And so from the rapid technological advantages in genome editing has made heritable germline editing a realistic possibility. So given the amount of research in the clinical trials that were presented throughout this article, I believe that gene therapy could give a lot of clinical importance. So gene therapies could possibly be the most complex drug developed, and they can potentially be a huge success. given the amount of expertise from pharmaceutical companies and the biotechnology that we have today, this article goes more in depth of the genome editing that is important to read and understand for the future, and try to find a good gene therapy to help cure or help maintain a disease. Thank you.